Very last thing we got is equilibrium. With equilibrium, we have what is equilibrium? It's this ability to understand where we are in space, most importantly, where our head is at. Because everything depends on where our head is at. We always put our head in a position that is parallel with the horizon. Okay. Just look at people. Okay. You'll see they're kind of, they'll have a slight little tilt to themselves because they have some imbalance, leg length. They've screwed up a joint in their ankle or in their knee or hip or whatever. But just look at people and you'll see that they're kind of, tilted a little bit. I'm over exaggerating it to kind of show it off. But their heads are never like this. Their heads are always like this. Okay. So even if you have a little bit of lean, the head always keeps it parallel with the horizon because this is how I'm going to interact. It's very hard to interact with the world when you look like this, when everything's slanted. So the body automatically positions your head to be in that equilibrium position. This is all monitored by what is called the vestibular apparatus. And the vestibular apparatus is the utricle, the sacula, which again is the vestibule, and our semicircular ducts, the canals. Okay. The fluid that moves within the membranous tube here of these three structures is what's going to trigger the receptor cells to allow you to know where you're at, where your head's at in space. Okay. Are you moving? Are you moving and not moving? What the hell does that mean? That's static equilibrium. Okay. If I'm standing still, but I'm traveling in an escalator, I am not moving, but the escalator's moving, right? And so by extension, I'm moving. That moves the fluid within the utricle and sacula for you to interpret that you are moving up. What if you're going down in an escalator? Same damn thing, just opposite. Okay. Also, linear acceleration. Okay. I am not moving, but let's say I'm in a car and it's all of a sudden makes a left-hand turn. Now that's linear acceleration. Okay. Or I'm standing still and all of a sudden somebody pushes me in my chair and I go zooming forward. Okay. I did not initiate that movement. Okay. Something else is moving. I'm just sitting in it. All right. There's your linear acceleration. The semicircular canals or the semicircular ducts, it's the membranous ducts within the canals. I get it. That's involved with dynamic movement, dynamic equilibrium, which is angular acceleration, because it takes into account the X, Y, and the Z axis. As you are doing the moving, you move, you move, you move. So again, this is where we're talking about, okay? This is where we're talking about. We're here in the vestibule. Here is the vestibule, okay? Right here. There's the vestibule, and then this is going to be, this is static, and this is the region for dynamic, okay? Here in the vestibule or within these semicircular ducts. So when we look at static equilibrium, I like to just call it static equilibrium. It's static equilibrium and linear acceleration. I just like calling it static equilibrium, guys, okay? For static equilibrium, it involves the utricle and the sacula, okay? The utricle and the sacula. Utricle is right here, and the sacula is right here. They are two distinct individual structures, hence two individual names. Inside the utricle and the sacula, though, is this thing called the maculae. Okay. And the maculae, there it is, located in, okay. This is right here on this right side, okay, over here. This is the maculae, okay? We have hair cells. Oh my God, just like the cochlea did. We have basically stereocilium, just like the cochlea did. And we have a long kinocilium, just like the cochlea did. Okay. So, I mean, really, it's the same setup, except now instead of sound, these things are going to move and we're going to interpret where we are. We have hair cells. Hair cells have stereocilia and one long kinocilium. In the cochlea, those little cilia are extending up and embedded in a tectoral membrane. Here, same thing. They extend off of the hair cell, but now they are projecting into another gelatinous membrane called the otolithic membrane. It is a jelly-like layer, just like the tectoral membrane, except this dude here has pebbles in it. And those are otoliths, okay? Tiny little stones, okay? That's what that means. They're actually calcium carbonate crystals. So when someone says you have rocks in your head, yeah, you do. There they are, okay? There is the otolithic membrane. The otoliths, tiny little pebbles, calcium carbonate crystals, embedded in that gelatinous goo. Okay? This is static equilibrium. Okay, so everyone just sit there. All right, look up. Just hold that position for a second. Okay, come back to center. Look down. Come back to center. What pulls rocks down? Gravity. So as soon as you bend your head down, gravity pulls those rocks, right? As gravity pulls those rocks, okay, it pulls that gelatinous membrane with it. When it pulls that gelatinous membrane with it, it bends the hair cells. Okay. So as you can kind of see here, look down, we are moving away from that kinocilium. And that causes a hyperpolarization. Okay. And we interpret that as looking down. If we look up, well, then it's just like we just saw with how we perceive sound, right? We bend kind of towards that kinocilium, and that kinocilium pulls on those links and opens everything up and depolarizes. And we interpret that as looking up. When you're just maintaining head in the upright position, not looking up, not extending, and looking up or down, those otoliths push straight down on those hair cells, okay? Pushes straight down on those cilia. As it's pushing straight down on that cilia, it's a stimulus, okay? It's pushing down. It's sending action potentials at a constant frequency in, okay? Again, gravity is pulling down on those stones, on those otoliths, okay? So it's exerting a pressure down on those cilia. And again, that sends action potentials. Now, again, when I look up, that causes a depolarization. That helps to excite and send more action potentials. So it's not just ions moving in or out per se, but it is now the rate at which we are sending action potentials into the brain. This is the first time we really see this phenomenon. We're going to see it in the next unit, though, as well. And we'll see it in the heart. So now as they bend towards the kinocilium as I look up, okay, gravity's pulling down those otoliths, right? Bending those cilia. This causes depolarization. So now even more action potentials are being generated. More EPSPs, if you will. Okay. So now the rate at which I'm depolarizing happens at a much greater frequency. And now I interpret as I am looking up. If I had you close your eyes and I came around and manipulated your head to look in the up position with your eyes closed, you could still tell me you were looking up or looking down because of how this thing works. Okay. Again, if I look down, okay, if I have my eyes closed, now I'm going away from the kinocilium. And whenever we go away from the kinocilium, we hyperpolarize. Okay. What does that do? Well, if we hyperpolarize now, there's even fewer action potentials being propagated. All right. For this, it gets hyperpolarized. It just means more potassium has to run through into the hair cell to get a depolarization. Well, that takes longer to do. So the frequency of action potentials is much, much less, to the point where you interpret that as looking down. Now let's just say I grabbed you. We were sitting in a classroom. I came, you were irritating me. I said, stand up. I picked you up by your feet and I held you upside down. Okay. What's gravity doing? Gravity's pulling down on those otoliths, right? But now instead of force coming down on it, force going down, now there's nothing being exerted down on those cilia now, right? You're upside down, for God's sakes. Gravity's pulling the other way. So now there's very minimal, if any, excitation at all. And you interpret that as I'm upside down. All of that is all that nonsense right there. Okay. As the head tilts, the otolithic membrane moves. That jelly otolithic gelatinous material moves against gravity that pulls those otoliths. And that causes a bending of the stereocilia. We bend toward it, we depolarize. We bend away, hyperpolarize. And that just changes the frequency for which information is coming into the brain to interpret where you are. Because these are always on. Okay? These are always on. These are tonic receptors, right? They're always bringing in information. You need to know where you're at all times in space, or you die. Okay? So you need to know where you are in space, and these are always bringing in information. Those otoliths are always being pulled down by gravity, always exerting a force on those cilia, always sending in action potentials at a consistent rate until you move. Okay? And then it depolarizes or hyperpolarizes depending on which way we're going. Towards the kino or away from the kino. What about movement? Just getting up and turning this way. That is angular acceleration, and that's what we got right here. Okay, I'm moving my head. Okay, when we have angular acceleration, dynamic movement. Okay, because this is dynamic, right? Me going up an escalator, I'm not doing anything. I'm just along for the ride. I'm not doing anything. I'm just in a mechanical device. Okay, that's not dynamic. I am not moving. I'm moving. Okay, so this angular acceleration is dynamic movement. When it comes to dynamic movement, I am moving now the fluid in my semicircular ducts, the fluid within those canals. At the base, at the very beginning, as you can see, we have this expanded region of every single canal. This enlargement is called the ampullae. And this is just a highlighted version of that ampulla right there. Inside the ampulla, we have hair cells, we have stereocilia. Oh my God, it's the same thing. Yep, it is. Except we call this entire structure the crista ampullaris. Why? Because we couldn't just keep it simple, right? Just call everything hair cells, everything stereocilium, everything kinocilium. We got to call things different, okay? So here, the hair cells and the stereocilia and kinocilium that extend off of it are collectively referred to as the crista ampullaris. Welcome to science. Those hairs, those stereocilia that extend off of those hair cells, okay, they are embedded into a gelatinous goo. Oh my God, it's the same thing. I'm just using slightly different words. Yes, it's all the same. The stereocilia are embedded in a gelatinous goo, just like it was in the tectoral membrane, just like it was with the otolithic membrane. The gelatinous goo here is referred to as the cupola. As the fluid moves within this semicircular duct, and again, that's endolymph, because this is the membranous canal, membranous labyrinth. The fluid will push against the cupola, and the cupola will move. Okay, again, if they were just there, hell, every time you just slightly moved a little bit, those hair cells would move in the water, right? Sending off depolarizations, you think you're moving all over the place, right? That would not be a fun world to live in. So that cupola helps to stabilize those hair cells. Okay, because as I'm sitting here, I have an inherent kind of movement that happens back and forth. Okay, and doesn't bother me. Okay. This doesn't really bother me a whole hell of a lot. Okay. The fluid that's moving within my cupola is very minimal. So that bends the fluid that's moving within the semicircular ducts is pushing against that cupola very little. So it only bends the hairs a little bit. I can handle that. Now, if all of a sudden I get on a tilt-a-whirl and I'm going around and around and around and around, now we're going to really bend those stereocilia and I'm going to feel like crap. Okay. But as fluid moves in within that semicircular duct, that pushes that fluid, that endolymph pushes against that cupola. And as it does it, bend towards it, depolarizes, bends away, hyperpolarizes. It's the same thing. So we have three different events occurring here, and it's basically all the same. Just changes the rate at which depolarizations occur within the vestibule or within the semicircular canal structures. There is a constant wave of depolarizations that are constantly coming in to let you know where you're at in the world and if you're moving. Okay. When you turn in one direction, that's going to cause those hair cells, the stereocilia of those hair cells, to bend toward the kinocilium, depolarizing it. Again, potassium is going to flood in the thing. And that increases the firing. That increases the rate of action potentials being sent from that crista ampullaris structure. And we will interpret that as if we are moving to, let's just say, at the right. Well, what if we're moving to the left? It's going to hyperpolarize them. Okay? And you'll interpret that as going to the left. There's data coming in from every axis, from the X axis, the Y axis, and the Z axis. Every plane of movement is covered here by these semicircular ducts. It is the ampulla that's found at the expanded end of each of these ducts. Within each ampulla, we have hair cells. We have stereocilia and kinocilium that extend off of them, collectively referred to as the crista ampullaris. These hair cells, these cilias, are embedded within a gelatinous goo called the cupula. As the fluid endolymph moves, when you move your head to the left or to the right, that causes the fluid to move, the endolymph to move within the semicircular duct. Pushing on the cupula, hair cells go towards the kino, depolarizes, increases the rate of depolarizations, increases action potentials coming into the brain, and we interpret that as moving to the right. Move to the left, we go away from the kinocilium, and we hyperpolarize. And we interpret that as we're going in the opposite direction. Takes time for that fluid to stop moving, right? Takes time for that cupula to bend back to the resting position, for those stereocilia not to be bent, and for you not to interpret that information. That takes a little time. How much? Depends on the person. The interesting thing, though, there are many different causes for vertigo. One of them is sometimes some of those otoliths can dislodge, and they can float within the endolymph. Well, as you can kind of see from that picture there, all of these tubes are connected, okay? And it doesn't really go back towards the cochlea. They always go up into those semicircular ducts. And if one of those otoliths gets up there, it moves the fluid, even if you may be standing still. Or it causes an exaggeration of fluid displacement when you move, which you feel like you're on a tilt-a-whirl then. And there's a procedure that you can do to help get these otolithic structures out of those semicircular canals and get them back into the vestibular structures. And it's, if that's the cause for it, it's actually, it's a pretty effective manipulation. So, and just watch the person's eyes. That's how you determine it. You can figure out, just watch their eyes when you got to put them on a tilt-a-whirl. And if they're really affected by the vertigo, it can cause them to really be sick. So you got to just take your chances with it. But it is a nice little procedure that is done, and it's very, very, very low tech. They do it in emergency rooms. I've done it in my office a number of times as well. So if that's truly the cause for vertigo issues, and you see that a lot with people who have head trauma, head injuries, elderly, boy, it clears it up right then and there. It's a pretty snazzy little move. When it comes to equilibrium, is a very involved type of sensation. There's a number of nuclei involved in here, so I don't probably a hint. I generally don't ask this one just because there is such a huge amount of input coming into the brain, not only from the vestibule and the semicirculars, but also the eyeballs and proprioceptive input coming from the joints, GTOs, spindle fibers. All that stuff is coming up in there, and the brain is interpreting all of those data points to understand exactly what equilibrium and head movement involves with maintaining your ability to be balanced with the world. So I don't necessarily ask it because it does so damn much. But you can see there from the vestibule, it can go all the way back, and it can really synapse with the cerebellum or the medulla oblongata. If it's medulla oblongata, it'll reflex with a number of different nuclei that are basically synapsing with the eyeball and bringing in information with the eyeball to help with your vestibule. Because as soon as I do this, my eyes are like, dude, you're leaning. So now I'm going to reflexively turn my body in this position to try to get me at equilibrium with the horizon. Cerebellum is just learned muscular movements, right? It's a blueprint for muscular movement. So obviously that's kind of why the cerebellum works in tandem with all of this nonsense. Got to have the cerebral cortex with the somatic motor cortex there to coordinate all these skeletal muscles. So there's a lot that goes into it. So I don't necessarily ask it just because probably go crazy trying to remember all that stuff. And I don't want you to go crazy just yet. Okay. But eyes, yeah. Sound waves, how it goes back to the cochlea to be perceived. Yeah, that's a good one. Okay. Equilibrium, not necessarily a pathway one that I like to ask. Way too many moving parts, way too many regions of the body.